Today we're going to talk about two chapters, chapters 7 and 8, that discuss different types of aquatic organisms and then a focus on microbes and plants. So if we think out to the three domains of life, the bacteria, the archaea, and the eukaryota, there are aquatic organisms in all three domains. The archaea are more different than bacteria than they are eukaryotes. And so keep that in mind. We used to lump bacteria and archaea together, but they're actually very, very different. And this idea of three domains replaces the five kingdoms you may have learned about in high school biology. Systematics is the study of both taxonomy, which is kind of how we name and organize organisms, and phylogeny, which is an exploration of the genetic relatedness among individuals. And so systematics takes both of these things into account. So here's another way to look at this, the eukarya. That's where we see things like some of the more complex protists, fungi, plants, green algae, and animals. The bacteria, we have things like E. coli, we have cyanobacteria, which are the blue-green algae. And then we have the archaea, which historically were thought of to be mainly extremophiles, but they're more common. So the species, how do we define a species? We can use what's called the biological species concept, which says that just a species is a genetically distinct group whose members can interbreed. But we hit some problems with this species concept in aquatic ecology because a lot of aquatic organisms don't reproduce sexually. So that definition falls apart. Additionally, there's a lot of hybridization, and that's when two species can interbreed. And so then where are the species lines there when we have hybrids forming? And many microbes are undescribed. And so we use the term operational taxonomic unit when we're talking about microbes because we don't have species names, we don't have them identified yet. And so we are more and more working with molecular data, DNA and RNA data to try to understand both phylogenetics and the taxonomy of a lot of aquatic organisms. Here's an image of the 16S region of the ribosomal RNA, and this is a molecule from a bacterium. The colored areas are universal sequences that are used to determine differences among organisms. And what's nice is that these areas don't vary much over evolutionary time. And so they help you to identify organisms. They have differences in these regions. There's also another problem with the biological species concept is that some species take on dramatically different forms. There's morphological variation. So this picture shows just one species of plant, but you see an emergent form with kind of rounded leaves and a submerged form with these feathery fern-like leaves. And so using the biological species concept, this plant was thought to be two different species for a very long time. Metagenomics is a tool that's coming online. We've been able to use it more and more frequently, and it allows us to sequence all of the DNA that we find in an environment. Really helpful for microbial work, work with aquatic bacteria, aquatic fungi, and really important for understanding things like the microbiome inside animals. So it's really great for microbial communities. And kind of a piece of this is using what's called eDNA, and that's environmental DNA. So it's basically going out to the environment, collecting all the DNA that you can find, and then determining what organisms may have been there in the past based on sloughed cells, feces, just kind of broken down bits of DNA that you find in the environment. It's really great for detecting rare species, and it's especially great for aquatic systems, and you'll read about that in the seminar paper to talk about why. Metagenomics is also really helpful when we're doing paleolimnology work, where we're looking at changes through time as we go down through ocean sediments or lake sediments. So here is another picture, this one from the book, that just shows you some of the groups of organisms that you would find And this is just another tree of life, but I think it just serves to highlight the vast diversity of the organisms that are not considered animals. So you can find animals down in the purple box in the group of pisciconts, and they're just kind of in there, like there's the animals, all of the animals, right? And then there's all of these other organisms that maybe you've not heard about. So this lecture will go on and tell you about the details about some of these groups. So there's the animals. Sorry. Another thing to talk about that's really interesting is this idea of what's called endosymbiosis. So we think that the chloroplasts of land plants were once free-living cyanobacteria. So you can see a picture of a chloroplast on the top. And we think that the mitochondria that are found in, say, our cells were actually once free-living purple photosynthetic bacteria. And this theory is called the theory of endosymbiosis, or the serial endosymbiosis theory, from 1966. And it was proposed by Lynn Margulis, who was a pioneering biologist. This idea that she had was not well received for many, many years. She had an extremely difficult time publishing this work, even though today it forms the foundation of basically all of modern biology. So it's a testament to perseverance and pushing through with evidence when you have it, even in the face of critics who don't believe you. So the different types of organisms that are out there, some of them are autotrophic, which means they're self-feeding, and you can be photoautotrophic if you use light energy, or chemoautotrophic if you use chemical energy. Other organisms are heterotrophic, which means other feeding, and basically they eat other things. And then there's a group of species that are mixotrophic, which can kind of switch hit. They can use both autotrophic and heterotrophic feeding strategies. Detritivores are kind of separated because they decompose dead material, organic carbon. They're sometimes also called saprophytes. So you would call them heterotrophic, but they're not feeding on living things. They're feeding on dead things. And then there are, you can organize aquatic organisms in particular into what are called functional feeding guilds. So the first are the grazers and scrapers, and they are invertebrates, insects, and fish that eat biofilm. Often the biofilm is made up of algae, but often it's made up of a very complex mix of things. Shredders are organisms that shred leaves and other organic material. Collectors that gather are organisms that wander around and feed on fine particulate material that's released by other organisms, things like feces and little bits of organic material. And that's a little bit different than collective filterers, who filter these particles out of the water column. And they can either do that passively by building some kind of a net, or actively by moving body parts through the water column. And then you have predators. And so you can either be a piercing predator or an engulfing predator. And predators consume parts or whole other invertebrates or vertebrates. There's also a whole bunch of really cool terms that are used to classify where organisms live. So benthic means on the bottom, epilithic means on rocks, epipelic means on on mud, versus epistamic, which is on sand. Hypereic is in the groundwater, but the groundwater that's influenced by the stream water, versus stygophilic, which is in the groundwater, or stygobitic. Then there's other terms like pelagic in the open water, and paraphytic, which is kind of like benthic, but it means, it comes from, well, we'll talk about it later, but basically a word for this complex biofilm on rocks. And then you should be familiar with some of these terms. It's basically, how do species interact with one another? So if you have two species, species A and B, and they can interact in lots of different ways, in ways where one is harmed, where one is benefited, where both are benefited, where both are harmed, and where neither is affected. Or, I'm sorry, if neither is affected, there's no interaction, but where one is not affected, the other could be benefited, or where one is not affected, the other could be harmed. So maybe pause the video while you fill out this table. See if you can name all of these interactions and their positive, negative, and neutral effects. Okay, Here is what you should have come up with. So mutualisms are positive, positive. Commensalisms are positive, neutral. Competition is always negative, negative. And then you have a suite of interactions where one organism is benefited and the other is harmed. So allelopathy, herbivory, predation, parasitism. And then the last one is amensalism, where one is harmed and the other is not affected. How'd you do? All right. So moving into the different types of organisms, one of the first people to kind of discover the diversity of life in aquatic systems was Antony van Leeuwenhoek in the Netherlands. And he is kind of a very famous individual in Delft in the Netherlands. When I was there, there are, placards and monuments, and you can go visit his tomb in Delft. There's a picture of him looking through one of his first invented microscopes, which gave him kind of a 300x view in the 1660s. And the first things that he saw were these, what he called animalcules. And some of these organisms you might actually be able to identify after this lecture and next week's lecture. So yeah, do you recognize any of these guys? You'll recognize them after you learn a little bit more. So when we think about the what's required for life, all organisms need a carbon source. We are a carbon-based life system on Earth, so you need some way to get carbon. You also need an electron donor, which is basically where are you getting your energy? And you need an electron acceptor, which is basically what are you breathing? And so for us, we get our carbon from our food, organic carbon. We get our energy from our food, and our electron acceptor is oxygen. We breathe oxygen, we breathe out carbon dioxide as a waste product. So autotrophs, they get their nutrition themselves. They make their own food, things like green plants, mosses, algae, autotrophic bacteria like cyanobacteria and some protists. So their carbon source is carbon dioxide. Their electron donor is light energy. And their electron acceptor, just like ours, is oxygen. They breathe out carbon dioxide. This is something that a lot of people get confused about. Heterotrophs, things like bacteria, fungi, invertebrates, their carbon source is like ours, organic carbon for their food for the most part. Their electron donor is often organic carbon. But there are a variety of electron acceptors. If you're an anaerobe, which is a lot of the organisms you're familiar with, they breathe oxygen and breathe out carbon dioxide. There's a whole bunch of organisms that breathe something else. So they can breathe CO2 or sulfate or nitrate or magnesium, iron, or sulfur. So here are some different life strategies. I would recommend taking a long look at this table and familiarizing yourself with, different types of organisms. They're going to come up in lectures that when we talk about ecosystem processes and the nitrogen cycle and methane and greenhouse gases and climate change. And so a lot of these organisms are really crucial in those processes. So there's actually two tables. So there's this one and there's this one. So lots and lots of really cool organisms that do a lot of things, and they get their energy and their carbon in a variety of ways. Okay. So I'm going to stop this first lecture here and move on to the different types of organisms.